Alright guys, we're going to do a quick little video lecture again on chapter 5 part 3, polypeptides of proteins. So proteins are the most diverse molecules found in all living systems, and the reason they're so diverse is because they're made of amino acids, and there are 20 amino acids found in nature that make up these proteins. So the possibilities are endless because you can have in any position, you can have up to 20 different amino acids. So let's say you have a protein that is 100 amino acids long. You could have in every position, so position 1, position 2, position 3, all the way up to 100, 20 different amino acids, which gives us so many different variations. So a couple terms that you want to keep in mind. Polypeptide is the same thing as a protein, so it's just the terms are used interchangeable. And then proteins have a specific shape, which then gives them their specific function. So proteins, it's obviously they're very important for living things. They're used all over the place in living systems, from bone, teeth, muscle. They're embedded within cell membranes. They're really, for the most part, the only thing that animals and plants can really make. So we take these proteins and we put them into a sequence that determines how we look, how we function, and what have you. One of the places you see them a lot is in enzymes, and we'll talk a little bit about that as we move forward. So as I mentioned earlier, that proteins come in a number of varieties, and the varieties are so different because each protein is made of a certain sequence of amino acids, and these amino acids are the monomers, or single building blocks. When we bind them together, we call them peptides. So you can have a monopeptide would be one peptide bond, a dipeptide would be two peptide bonds, and then a polypeptide would be 3 or more of those bonds. So once again, I'm going to fast forward a little bit here so we can talk about the structure here. So if you look at all of these are all different amino acids, and these ones happen to be hydrophobic, meaning that they're water-fearing. And if you look at the shape of them, on one side there's an amino group, there's this carbon in the middle, and then there's this group on the other side, the acid group. What they're showing you here is that the negative portion is the H has left the hydroxide ion, which would normally be OH, and has been picked up by the base. And bases tend to pick up hydrogens, and acids give away hydrogens. The white structure that you see there on each one of these are what they call the side chain, or R groups. R groups are the things that are different in every amino acid. So if you look across the top here, you see NH3CHCOO, NH3CHCOO, NH3CHCOO. Hopefully you see that structure stays the same the entire way, but these groups, these side groups or R groups, change depending on which amino acid that we have. And finally, these amino acids here, they tend to be, these ones happen to be hydrophobic or nonpolar because they don't have any polar characteristics. Notice there's no charges. Oops. There's no charges here. There's no oxygens. There's very few nitrogens. All those things that make things polar. A couple of these should look familiar. Hopefully, let's see. Tryptophan. You might know this one. This is what's found in turkey. It's what makes you tired. Asparagine, which kind of sound like asparagus because this is what's found in asparagus, this particular amino acid. It's what gives asparagus that kind of a odor, if you will. So there are also hydrophilic R groups. So notice these groups here, the top stay the same as before, but as these groups change, we have OH, which are polar, OH, which is polar. We know that NH2 is basic. Basic is polar, and SH is similar to OH, so that's polar. There also are acidic and basic functional groups, and this kind of rounds up the 20 different amino acids. Acids, remember, give off hydrogens. That's why they have OH minus. It's kept onto the electron that used to be part of hydrogen. And we have bases, which are going to be NH3 plus. Normally it's NH2, no charge, but it picked up a proton, which had no hydrogen, which is here, here, and here. Okay, so how do these things work? So you can see the purple structures here. Each one of these purple structures represents an amino acid. Here we have amino acid number 1, which has its amine group and its acid group, and its amine group and its acid group, its amine group and acid group. The way they bond together through dehydration synthesis. So we have a hydroxide ion here, we have a hydrogen ion, and we drop those two. We drop those two to produce water, dehydration. A new bond takes place between the C and the N, the C of one amino acid and the nitrogen of the next amino acid. This is called a peptide bond. So this structure here with two amino acids would be considered a monopeptide because it only has one bond. This would be a dipeptide because there are two peptide bonds. Okay. So this is pretty neat. So what we've done here is if we take a string of these amino acids, I'm just going to show you this in a different way. So here's a string of amino acids. Amino acid 1, 2, 3, 4, 5, 6, position 10, position 15. So these are all these amino acids that are all strung together. When you string them together, you get what's called the primary structure, or the sequence of amino acids. And the sequence of amino acids is important because the sequence dictates how the protein will then wrap and fold on itself. And it's going to wrap and fold on itself to make these three-dimensional shapes that then have a certain function. So let me go back again. So here are a couple pictures of proteins in their three-dimensional shapes. Here's a space-filling model. Here's kind of a ribbon model, they might call that. Okay, and the shape of these dictate how they work. I'll use myself for example. I am lactose intolerant, so I don't make the enzyme lactase, which digests the sugar lactose. So because of, and the reason for this is because the enzyme that I currently make for lactase is the wrong shape and doesn't fit around the lactose sugar. That being said, I don't have the ability to digest that type of food source, so it kind of runs through my system. It doesn't really get digested, which is not a good thing. So once again, so there are three levels of protein structure. There's primary, secondary, tertiary, and these are how we'll kind of explain how each one of these work as we go. So once again, primary structure, we just looked at this picture a minute ago. This here shows 129 amino acids chain. All right, so it's amino acid 1 stuck to 2, stuck to 3, stuck to 4. So the sequence of these amino acids is very important. Humans, for example, I'll use blood types. We have A, B, O, and AB blood types. Really, there's only two. It's A and B. Of those, and the reason there's only two is you either make A proteins, you make B proteins, or you make no proteins. If you make an A protein, there's a certain sequence of these amino acids, and every human that has A blood type has the same sequence. Therefore, their A blood type proteins, or their A blood type, is transferable to anybody with A. B blood type makes B proteins, not A. The sequence of amino acids is different, so because that sequence is different, B can only give to B. O blood type people don't make a protein on the outside of their cells, so they can't, they don't have this protein here that gives them a specific sequence. So therefore, those people can only take O blood because they have issues with getting A or B. And AB obviously make both A and B proteins. So once again, primary structure. If it's not the correct primary structure, if you're in this case the 129 amino acid chain, if it's not correct, we get misshapen proteins. When they get misshapen, here's an example. This is an example of sickle cell blood cells. Sickle cell blood cells have this sickle shape here, and the sickle shape causes them to stick and get caught in people with this disease's capillaries. The other thing that's a benefit with it, though, through evolution, is that people that have sickle cells don't get malaria, which you find in sub-Saharan Africa. All right, last couple real quick. We have secondary structure here of a protein. They're either sheets or beta sheets, which are here, or helixes. All right, these shapes are formed when the hydrogens on the backbone here, these hydrogens here, at the very top, when these hydrogens along this backbone interact with one another, and you get either sheets or coils. These are things that actually keep that make protein strong. We see it in spider webs and places like that. All right. The last type of interaction is here, tertiary structures. These are structures that the R groups or side chains bond together. So things that are like bond together. So here we have sulfur bonding together. Here we have things that are hydrophobic bonding together. We can make hydrogen bonds. There are ionic bonds, plus-minus bonds. These are the type of bonds that then cause, we talked about hair being curly. If you have a lot of sulfur in your hair tends to be curly because it makes these loops like this. Okay. Let's see. Quaternary structures, each one of these blue lines or orange line here represent a single protein chain. If you take multiple protein chains and mix them together, you get this quaternary structure, which we'll discuss a little bit more in class. So here's kind of the situation. We go from primary to secondary to tertiary to quaternary structure. These all have can be affected by things like heat, temperature, pH, and salt concentration, which we'll talk about in class. Hope this helps. See you in class.